you. Okay, so how was the vacation? Great. אי אפשר בלי הטלפון. Okay, so again this time the reminder I think is more important because it's been a long time since the last class. So I will not go back to the chapter of the like the cell architecture. You had questions to answer about that, but we will go over again briefly what we did already in the chapter of transport of ions and small molecules across the cell membrane. So we started this chapter by saying why would the cell need to transport anything into its membrane, and the obvious reasons is nutrition for the cells to get like the nutrients that they need to function. One that is very important to neurons, way of transportation is in a way to like convert chemical energy to potential energy. So we talked about the differences in ion concentration between the inside of the cell and the outside of the cell. And these differences are actually harnessed by the cells as available energy. Probing and signaling to sense the environment or to get information from the environment. For example, neurotransmission in neurons. So they need to transport several molecules that help for this transmission. And in some cases, cells can also change their volume. So they can use water. Pump in water or pump out water to change the volume of the cell. And so we first said that not all molecules, like obviously the barrier of the cell membrane or the plasma membrane of the cell is not completely impermeable. Like there are molecules that are able to freely move across the cell across the cell membrane. I touched that. For example, small gases that are not charged, like oxygen or carbon dioxide, can freely move across the membrane because they're uncharged, and we said that this is like a hydrophobic barrier. So small molecules that are uncharged, like gases, can pass through the membrane. Also, other small molecules that are uncharged, like ethanol, can penetrate the membrane relatively freely. That's why you get drunk very easily. Also, in that note, if you know of a lot of like etheric shmanim etherim or etheric oils or essential oils that you have from plants, a lot of times they're very potent because they can pass through membranes very easily. For example, you can try the experiment that you put, but don't put it too long. You can put like a slice of garlic on your toenail, and then like after an hour, your breath will start smelling like garlic. And this is because the etheric, like the essential oils in the garlic and also in other plants are very, very potent and very permeable to cell membrane in general. So you can imagine, for example, for ethanol, your body is like this huge sack of liquid. So the ethanol just disperses along your blood system and everywhere because it's just permeable to all the membranes. And for other substances, it's very compartmentalized. I don't know. Like my diction in English is not that good. So yeah, it's divided to compartments. Yeah. And okay, so Water is slightly permeable because it's small, but it's polar, so it's slightly permeable. So it means that under normal conditions it will normally not pass, but if there is high osmotic pressure, like water pressure in one side, they can still pass some of the molecules. And then all the other molecules, other large molecules that are uncharged, or molecules or small molecules that are charged, are impermeable to the cell membrane, meaning that the cell has to actively transport them through the type of channels that we learned about. There's something. Okay, I'm not going to touch this anymore. So basically, when we can divide transportation across the cell membrane into three categories. All of these are proteins that are transporting molecules across the cell membrane. And all of these have to be obviously transmembranal. They're not like on one side of the membrane and the other side of the membrane. And all of them will have a hydrophobic domain and a hydrophilic domain. The hydrophobic domain will be inside the membrane, and the hydrophilic domain will be outside the membrane. And you can either have pumps that are in charge that can pump a molecule against its concentration gradient by the power of ATP, or you can have ion channels that they can either be like freely transporting, or like gated or non-gated, but it still doesn't really matter because they don't need energy to transport ions across them. And you can have transporters that some of the transporters are active, some of them are passive, similarly to the ion channels, but the only difference between this uniporter, for example, and a non-gated ion channel is that here there's a conformational change that's happening for each and every molecule has to pass like a conformational change in order to pass from here to here. And in the channel it's just like a hole, basically, that freely passes. And from this you can see the difference in kinetics. So here, this is much faster than this. This is the fastest way of transportation because it doesn't require energy or conformational change in order for the ions to pass. So we gave specific examples. We talked a little bit about the concentration gradient and the forces that are in charge of the concentration gradient. Like in the end, everything that we're learning about, the forces that are driving all the phenomena that we're learning about is entropy and the interaction with water and entropy and all this stuff. We didn't get into it in very much detail. But still, in the examples of ATP-powered pumps, we gave the example of the P-class pump, and we talked about it in two instances. One, in the case of the sodium-potassium. So we said that there are concentration differences, and I'm not going to repeat that, between the inside of the cell and the outside of the cell. And that these pumps require a lot of energy. For example, 25% of all the ATP in a nerve cell is used to maintain this gradient. Hence, these kinds of pumps that are in charge of maintaining these differences between ion concentration. So we gave the example of the sodium-potassium pump, which is a very famous example in neuroscience, which is in charge of maintaining a high concentration of sodium outside the cell and a high concentration of potassium inside the cell. We gave the example of calcium from muscle cells. That calcium is the one that is in charge of contraction of muscles. So in this case, there's a difference in concentration between what we call the special organelle that is existent in muscle fibers, which is called the sarcoplasmic reticulum, which are like internal sacs that store high concentrations of calcium. So the SR, sarcoplasmic reticulum lumen, is highly concentrated in calcium, and the pump that is in charge of that is again a P-class pump that transports calcium from the cytosol to the lumen of this organelle. And this is like a molecular model of this organelle. What is important for you to see here is that this conformational change is something very drastic. Okay, there's when you look like location to location between these two, between what happens when this channel is either open to the lumen or open to the cytosol phase. So there are massive conformational changes that happen between this change and this state and this state. So it's not like a simple, it's a very complex mechanistic thing that's happening structurally in the protein in order to allow this, the movement of these ions. And we started talking about, we actually talked about non-gated ion channels and resting memory potential. So we said that the concentration difference, like the P-class pumps, are responsible of creating the concentration difference between the inside and the outside of the cell. But the channel that is responsible for the electrical potential, okay, so the electrical difference, which is a thing that's different from the concentration difference, is a resting, what we call the resting K. We gave the example I'll show you in a second, which is a non-gated ion channel, which basically allows free flow of ions through this channel. But what's special about this channel is that it has a selectivity filter, or this is this segment, it's a tetramer. So this protein is a tetrameric protein, which means it's made from 4 different subunits, or in this case I think they're identical subunits. But what's special about this structure is that it has a selectivity filter that only allows for potassium to pass. So we gave the example that although sodium is actually much smaller, so this is like the atomic weight of sodium, and this is the atomic weight of potassium. So although sodium is much smaller than potassium, and they have the same charge, still sodium can't pass, because when sodium is in an aqueous environment and when it's hydrated, it interacts in water in a very, like the interaction with water creates like this complex. And if the sodium would have to pass through the pore, then the interaction with the oxygens inside the channel pore would mean that it only will interact with 2 oxygens, actually 4, but we only see 2 here, but with 4 oxygens out of the possible 8. So this interaction is much more stable than this interaction, and this is why the sodium would prefer to be in the water than to be in the pore, although it's smaller. And for the potassium, it's actually like a smooth transition. So there is almost no energetic difference between this state and this state inside the pore, and this is why the potassium can pass through this pore and the sodium can't. Question about that? Okay, so, and this is another illustration of that, but I think we get the point, I hope at least. And again, to demonstrate to you in a very simple way how this resting K, or the permeability, the selective permeability of a specific ion, can help us generate a membrane potential. So we gave this simple example, where here you have a concentration difference. For example, the sodium again is very high here, and the sodium here is very low. Again, these are the concentrations. This is 15 millimolar and 150 millimolar. But still, because all the ions here are balanced with chloride, then there's no electrical difference, because there's no difference in charge between these 2 compartments. But if we induce a permeability to potassium, then what will happen is that potassium ions, because there's a high concentration here, will start flowing in this direction, and eventually, after enough potassium ions will flow in this direction, because now this side is now positively charged, then there will be a force, like an electrical force, that will start pulling the potassium ions like in this direction, like in the opposite direction. And in the end, there will be a resting state in which enough, like the potassium ions, the differences in concentration will be that this is what we call the electrochemical steady state, or the electrochemical potential, in which in one side the force, the chemical force, is pushing the potassium ions in this direction, and the electrical force will push the potassium ions in this direction, and and the balance in what we see in biological cells is that the memory potential will be about -60 inside the cell relative to the relative to the outside environment, and minus 60 is millivolts, or minus 60, minus 70, something in this area, and we can explain the entire potential only by allowing permeability of a single channel. Okay, but still the concentration doesn't change because the number of channels is very small relative to the number of pumps that keep maintaining the concentration constant. Okay, I know it can be like confusing because you can see something permeable and something, but still the concentration remains. But this just results from that there is a huge amount of pumps that are in charge of this concentration, but only a small amount of channels that allow permeability of potassium. Okay, so this is like a steady state in the cell. Okay, so we also mentioned the third type of transport, which is transporters. So again, this family are grouped together because they require conformational, because they don't take their energy from ATP, so they're not ATP powered pumps, but they do require conformational change in order for the ions to pass. So the most simple transporter is a uniporter, in which is passive, and it's passive because it doesn't require energy. So the ion or molecule that has to pass here is just passing down its concentration gradient. And in the cases of symporters or nandiporters, we will say that these are active transporters because they harness energy, and in this case from the red molecule that is going down the concentration gradient in order to transfer another molecule up its concentration gradient. So it can either be, if these two molecules are traveling in the same direction, then we will call it a symporter, and if the two molecules are transported in the opposite direction, we will say antiporters. Okay? And I gave you a classical example of how the cell uses these antiporters in order to maintain the pH. And a point that I think I didn't stress enough before is that the way that the fact that these transporters need to undergo conformational change means for us, for like people that study structural biology or something like that because their activity is very much dependent on their structure. So the protein has to have a very, very specific 3D structure in order for it to be active. For example, you can think about if you have like a motor that has a small dent, like somewhere in the cylinder or something like that, then it will not function. Okay, it will still look like a motor, but it will not function. So proteins are actually very sensitive to pH, and these transporters are actually designed. I don't know who designed them, but they're designed in such a way that in each pH you have a transporter that will be more active than the other. And how does this happen? It happens exactly because the pH determines the level of denaturation of the protein. So pH affects directly the structure of the proteins. So some of the proteins, like for example this transporter, the chloride HCO3 transporter, will be active in this pH, meaning that its ideal structure or its ideal 3D structure of activation will happen in this pH, and this transporter, its ideal structure or the ideal 3D like folding and morphology of this protein will be in this pH. So and this will not be active at all because it's like the structure will be inactive. So it's kind of I can't even find examples for that in our life or metaphors for that in our life because it's so different from concepts that I don't know that you can think about, that the pH, while it changes, it actually changes the structure of each one of these different transporters, and each pH is and by this changing of the structure, it actually changes the activity of the transporter. So so this is I think this is the reason that this is not like a simple channel or something like that. But last class I did talk to you about the kinetics and exactly give you an example of what happens in each time, but it's easy to see if you know a little bit of dynamics that the fixed point or the steady point will be here. Okay, between the, like there will be a transporter that if the cell is too acidic, it will push it towards this direction. But when the pH arrives in this range, then the dynamical or the dynamics of these two transporters will drive the pH to be in this point, which is not surprising to be the physiological pH of the cell. Okay, so we said really a few words about aquaporins because they're not very interesting. So basically, they're just channels that transport water. There's not a lot to say about that. And we started talking about voltage-gated ion channels and the propagation of action potential, although I'm really neglecting the propagation of action potentials because, again, I assume that you learned about this in other classes. And okay, and this is the last place I think we stopped. So you had questions on the last chapter, which is biomembranes and cell architecture. And so we'll go over them very quickly. So some organisms in the cell are surrounded by two membranes, and in between these membranes there is an intermediate space. Which are these organelles? So, and when I mean two membranes, I don't mean the two leaflets of the membrane. I don't mean like the plasma membrane. I mean two membranes, like four leaflets, if you like, really a double membrane. Okay, so is it the Golgi in the lysosome? Is it the nucleus in the mitochondria? Yes? So to remind you, because it looks like you obviously didn't go over the questions, so why am I? Actually, the animations here are not working. Anyway, the nucleus in the mitochondria, I'll go back to it. Okay. So the nucleus and the mitochondria, like you see here, they have a double membrane. Okay. So we talked about the nomenclature of a lumen, which is the internal space that is not the it's an internal space of the organelle. So this is a single membrane, again, like the membrane of the cell, and here there is a double membrane in the nucleus and in the mitochondria. Okay. So you said correctly that in the nucleus and the mitochondria there are double walls. Yeah. So, and here the animation doesn't work, so it's even better. So below is a list of cellular organelles. Match the organelle or one organelle or more from the list to its role in the cell. So these are the organelles, and this is the function of them in the cell. So the organelle that's responsible for the degradation of outside material and all cellular organelles is the lysosome. The organelle that is responsible of ATP synthesis and glucose oxidation, which is actually ATP synthesis, is mitochondria. Synthesis, processing, and sorting of secreted proteins is the RAF-ER, and also in the Golgi apparatus can be. And storage of secreted proteins happens in the secretory vesicles, and processing and sorting of secreted proteins happens in the Golgi apparatus. Okay, so this you should know even from this class. Integral membrane proteins are usually, so mark all the correct sentences, are they rich in alpha helices? Are they rich in beta sheets? Are they rich in double helix structures? Where does the double helix structures come from? DNA, so it has nothing to do with this. Are entirely hydrophobic?
No.
Contain a hydrophilic region?
Yes.
Contain a hydrophobic region? Okay, the animation doesn't work. But yes, it's A, E, and F.
No.
Ah, now it works. It's the opposite. Okay, good. So am I going in the wrong direction? No. It's very strange. Anyway, so voltage-gated ion channels, which are very important to neuronal functions, obviously. I'm not allowed to lean on this anymore. Okay. So voltage-gated ion channels are very important for neuronal functions. Like their name implies, they have a capability of sensing the memory potential, or sensing the differences in voltages, or the difference in charge between the inside of the cell and the outside of the cell. And once they sense this change, or the change that is suiting for this specific channel, then they have this gate. And obviously this is a wild simplification of this gate. But for example, if we're looking at the basic model of an action potential of a neuron, then again, this is the concentration difference, and always remember that this is a constant thing. Okay, it doesn't change, even during an action potential. And this is always open. Like we said, the resting K channel is always open. It's non-gated. But there are a lot of sodium channels that are closed, and they're gated, so they have this block. But these sodium channels have the capability of sensing differences in voltage, and when there is a change, or a change in the polarity of the voltage between the internal part, the exterior and the internal part of the cell. So you see here there's positive and here is negative, and here in this condition it's flipped. What will happen is that this gate will open, and it will allow free flow of sodium through these channels. So again, we will not say that this is an active channel, because it doesn't require ATP, but it is gated, so there is a small conformational change that's happening here, but it's not directly linked one-to-one to transport of the ions. It's more like a gate that's opening, and the ions freely flow through it, to cause the cell to be even more depolarized, to be even more positive in the internal part of the cell. And there are a lot of different models. Actually, you have for some of you that are interested, you have in the Moodle like a small review article that shows like the different mechanisms of voltage sensing, like how proteins sense voltage. And this is just one of these models, but it's a very typical model. It's very common also in voltage-gated ion channel, in sodium voltage-gated ion channels, and also in potassium-gated ion channels, is that somewhere in the 3D structure of the protein, or the third structure of the protein, you have this positively charged alpha helix that is sitting in the core of the protein, and this alpha helix is what we call this voltage sensing helix. Okay, and it's voltage sensing, and again, it's rich in lysine and arginine side groups, which are positively charged. And you can see here that, for example, in this state, where the cell is in resting state, so the internal part of the cell is more negative than the external part of the cell, then this voltage sensing helix will be drawn towards this direction, or to the cytosolic phase, and this pore will be closed. Okay, and what happens when you have a depolarizing signal, or that the potential, or the polarity of the cell changes from inside the cell to a negative to a positive polarity, then this voltage sensing helix will move. This is also obviously a massive conformational change that happens inside the protein, and because of this movement, there is a channel that opens, and sodium can now freely pass through this channel. Okay, because again, there's a high concentration of sodium outside the cell and a small concentration inside the cell. But in this case, there's also this special type of ball, or what we call a globular domain of this channel, that can actually inactivate this channel a very short time after it's been activated. So this inactivation domain, or this globular bolus domain, is what causes this channel to only allow movement of ions to a very brief amount of time, and then this ball moves to this position and actually physically blocks this channel, and this channel becomes inactive. So it's actually, people think it's like the kinetics. It just takes time for this domain. So once this domain also like senses the voltage in some way, and just when the voltage is, when there is depolarization, then it already starts the movement towards this direction, but it takes it a little time to get here to block the channel, and during that time the sodium is moving freely. But it's actually very complex. Like it's part of the things that we don't really know. There's like there's a lot of like different models to describe how this gating works, or the gating itself, and also the inactivation. So this is the simplification, but it's a nice model anyway. So so you see here the inactivation happens in half or 1 millisecond. And the depolarization itself happens less than 1 millisecond. So there's like 0.4 to 0.9 milliseconds when the channel is open before it gets inactivated. So molecularly, if we compare two types of, if we look in the structure of these voltage sensing channels, so here you have a nice comparison between a sodium voltage gated channel, which is the same channel that you saw before. So this is this channel. Just you can see here now in a different layout because we want to see different things. And a voltage gated potassium channel. This is not the same channel that I showed you before. It's a voltage gated potassium channel, and you can see that structurally, actually the only differences, the major differences are shown like here in blue. The structure of these proteins, or the amino acid sequence of these proteins, is very much similar to one another. So they're very homologous. But there's one major difference in which, in the voltage gated sodium channel, this whole protein is a single monomer. So it's like it's made from a single gene. So if we return again to the layout of how a gene looks like, so there is a single gene that will code for the entire protein. Okay, and you can see that this is one end of the protein, and this is the other end of the protein. This is the amine end of the protein and the carboxylic end of the protein. And here we will say that each one of these replicates, or which are pretty similar to one another, these alpha helixes, that alpha helix number 4 is the voltage sensing helix. We will say that these are 4 different domains of this protein. And in the case of the voltage gated potassium channel, each one of these are actually they're actually subunits and not domains, because each one of them is coded from a different gene, meaning that, or from a different gene, or from the same gene, but like it requires several copies of that gene. Okay, so here you can see like a few differences between these two channels. One of them is in blue you can see the area of the selectivity filter that is responsible for this channel to only transfer potassium, which is you see it's very different from the sodium one. And also the voltage gated potassium channel has these 4 globular domains, globular inactivation domains, and the sodium has only one, which is not here. It's just like H. You don't see really how it looks like. Okay, so it's clear that in this case, like it looks like these two proteins are very similar to one another, but actually here we will say that there are 4 subunits. We will call this protein a tetramer, and here it's the same gene that encodes for all these 4 domains, and this is why we will say that this is a monomer. Okay, so it's very important in terms of nomenclature. But you can see that in structure they're very similar to one another, with slight changes between them. So a few words about the voltage gated potassium channel. This is like a simplification again of the structure, and this channel is made from it's a tetrameric protein, like you saw before, and it's made of 4 identical subunits or alpha subunits, and other 4 beta subunits, but we're not talking about them right now. And each one of these alpha subunits has this globular inactivation domain. And one of the experiments that this channel is actually called a Shaker K channel. You will see that each time that you have a protein that has like this cool name, that's not like NHBA1 or something like that, when it has a real name, it means that it was discovered in Drosophila. Because I always say that Drosophila people have a sense of humor, and all the other don't really have a sense of humor. So the reason it's called Shaker K is because the fly, or the Drosophila fly that they mutated this channel, had a phenotype that it was shaking. Okay, so when this channel was mutated, then the fly would shake, and hence the Shaker K. And actually, the mutation that they did was to delete this inactivation domain. So in experiments, when they try to measure the kinetics of this channel, so the wild type or the normal activation is that when there is depolarization in the cell, then there will be an influx of potassium. You see this red or purple red thing. So there will be an influx of potassium actually to the outside of the cell. And but very fast there will come an inactivation. It actually happens much slower than the sodium, but still relatively fast. There will come inactivation, and gradually the influx of calcium will decrease in the wild type variant. But when they mutated this channel and actually deleted the inactivation segment, you can see that once there is a depolarization, then the channel opens and it stays open. Okay, there is no inactivation. And again, to prove that this is really the changes that they did, they added a synthetic peptide that mimicked the structure, or a synthetic protein that mimicked the structure of this globular domain, and you can see that they could rescue some of the original activity. So this is like this is the mutant K channel where this green thing is missing, but they added like a synthetic peptide that is supposed to replace the function of this globular domain. Okay, so I always tell you that. So let's see if you get one of the messages that I'm trying to deliver here. So nerve cells can conduct many action potentials in the absence of ATP. This is like a fact. Typically, there are 10 voltage-gated sodium channels per square micrometer of plasma membrane in the normal plasma membrane of the cell. In the axon hillock, where the action potential is generated in neurons, there's about 100 to 200 voltage-gated sodium channels, and in the nodes of Ranvier, which are the areas along the axon where the action potential is regenerated, then you have 1,000 channels per square micrometer. So each channel passes about 5,000 to 10,000 ions during the millisecond it's open, and overall there are 2 million sodium ions that enter and 2 million potassium ions that leave during an action potential. And what I'm asking is that, again, a lot of people confused because they say all the time that there is opening, there is entering of sodium and exiting of potassium. So my question is it a lot? Is it significant? Or in other words, is it enough to change the concentration of the ions in the internal part of the cell and the external part of the cell? No. So yes. So it's not a lot. And actually, during a typical action potential, the number of sodium ions in the cells increased only by 0.05%, something like that. So the action potential doesn't change the concentration of the ions, but the movement of the ions is enough to cause a fluctuation in the membrane potential. So again, there are two different things. The concentrations and the membrane potentials are something that are, it looks like they're coupled, but they're actually not. Okay? So, where are we? Yeah. So, the last chapter, the last part of this chapter, we're going to talk specifically about neurotransmitter and receptors and transport of proteins in transmission in synapses. So exactly what happens, how is all this transmission happening in synapses? So first, neurotransmitters are mostly made of, are derivatives of amino acids, and sometimes they can be also derivatives of nucleotides. For example, ATP, which lacks phosphate groups, can also be a type of neurotransmitter. So these are just examples of different types of neurotransmitters that we have in the central nervous system. So acetylcholine, glycine, glutamate, which is the central activating neurotransmitter. Dopamine, which I'm sure all of you heard about. Norepinephrine, which is again important for a lot of neurological symptoms. Epinephrine, serotonin, and histamine. Actually, I don't know where it functions. Histamine is a neurotransmitter, you know. Yeah, but in terms of neurotransmission. So all of these are examples of neurotransmitters. I don't know, maybe in the immune system, but the last neurotransmitter that I'm gonna mention is GABA, which is actually the major inhibitory neurotransmitter of the central nervous system. And so, how exactly does neurotransmission occur? So the first step of the cell, in order for it to release a neurotransmitter, is to pack it inside these small vesicles. So, and it does so in two steps. The first step is more actually, we'll say a few words maybe before that, in which more general about neurotransmission. Actually, like in past presentation, I would say that each neuron generally produces just one type of neurotransmitter, and this year I actually changed it to actually not true anymore. So this is one of the things that we learn in neuroscience. Nowadays, it's more or less, we know of a lot of different types of populations of neurons that actually co-release two types of neurotransmitters and not restricted to one type of neurotransmitter. It's still very much like it's not unknown what is the function of this double release or how it exactly happens, but we do know that there is that in many types of neurons they actually release two types of neurotransmitters. And also this is not true anymore. Synthesized in this is actually true. Synthesized in the cytosol and imported into membrane-bound synaptic vesicles. So again, it happens in two stages, and next class we're going to get into detail exactly how this happens. But these vesicles are 40 to 50 nanometers in diameter, and their lumen has a very low pH, that this low pH is generated by V-class ATP pumps. So we talked, I showed you one example of an ATP-powered pump, which is the P-class pump, which is a sodium, potassium, and calcium pump. And this is the second type of ATP-powered pump that we're going to talk about, which is the V-class. And this pump, you can see that it has a very, very complex structure, is in charge of hydrolyzing ATP, and each hydrolysis of ATP gives the energy to transfer two protons or two hydrogens inside the lumen of the vesicles. So the difference in pH, for example, because you obviously know by now that pH is the same as, say, the inverse concentration of protons or hydrogen ions. So the pH inside the lumen, or the space inside the vesicle, is like, for example, 5.7, and the pH outside of the cell is 7.2. It's like the normal physiological pH. So actually, this acts in a very similar way to like the cell. So I told you that the cell has several ways that it can store and use energy. Okay, so the pump that I showed you in the past, like the sodium potassium pump, converts the ATP and gives the cell the concentration gradient. And this concentration gradient is then used as an energy source for the cell for a lot of different functions, for example, to drive an action potential. Okay? So in this case, it's very similar because the cell or the cell uses the ATP in this case to pump hydrogens inside the lumen and create a large concentration differences of the hydrogens from the inside to the outside. Later, it will use this concentration difference using in this case, an antiporter. You see that that will take one hydrogen outside, like with its concentration gradient, and harness this energy in order to pump the neurotransmitter from the cytosol or of the cell to the lumen of the vesicle. Okay, so it's like a two-step process. And for example, one of the, we'll talk about it actually after the break because there's a lot of things to say about that. Okay, so let's take a break and we'll return to that next class. כי יש הבדל מאוד קטן בריכוזים, זאת אומרת ההבדל בריכוזים מאוד קטן, מה שמשנה כאילו... מספיק להפוך את פוטנציאל הממברנה? זהו, אז צריך מעט מאוד איונים בשביל להפוך את הפוטנציאל הממברנה. לא, זה קרוב לממברנה? לא, זה לא עניין שזה קרוב לממברנה. זאת אומרת, מה שהראיתי לכם פה, זה מסביר את זה בצורה... פה. אוקיי, אז כאילו... לא, זה בדיוק זה נורא קשה אינטואיטיבית להבין את זה, זה לא היה הגיוני, אבל זה עניין, תחשבי שנגיד, פה יש הבדל ריכוזים מאוד גדול, כי פה יש סודיום, כאילו סודיום נמוך וסודיום פה גבוה, אבל אין שום הבדל בפוטנציאל, בגלל שיש פה כל איון, כן, השמאלי אותו דבר, כן, מספר המטענים החיוביים השליליים פה ופה, כאילו פה הוא שווה ופה הוא שווה. עכשיו, אם אני רק אכניס אני אעביר כמה מטענים חיוביים מפה לפה, אני לא צריך להעביר הרבה מטענים חיוביים מפה לפה, זאת אומרת, זה לא יהיה מספיק לשנות את הריכוז באמת, אבל המתח החשמלי ישתנה. זאת אומרת, זה כאילו, זה קצת עובד כמו סוללה. זאת אומרת, סוללה, זה בעצם עובד בדיוק כמו סוללה. זאת אומרת, בסוללה, את משתמשת בסוללה במשך הרבה זמן, נכון? כי אם היית כאילו... נכון, זה היה מספיק לשנייה, כל המטענים היו עוברים לצד שני וזהו. אז בסוללה יש מעבר של מטענים, אבל באופן כללי הריכוז, אם הסוללה היא גדולה מספיק, וגם חושבים שהסוללה הייתה נטענת כל הזמן, שזה באמת מה שקורה פה, כי יש כל הזמן טעינה של הסוללה, אז את מקבלת את המתח רק ממעבר של מעט מטענים כל פעם. ואז המטענים האלה זוגים, עד שבסופו של דבר הם משפיעים בסוללות. אבל נגיד אם הסוללה באמת שנטענת כל הזמן, אז כל הזמן היא מקבלת מתח, אבל הריכוז של האיונים היה נשאר קבוע. זה באמת קשה להבין את זה אינטואיטיבית, זה כאילו לא... אבל זה פשוט ככה.
want to copy the whole URL, or you want to just
Let me see first if it
just do a search monotone in their website. It's not melatonin.
מה זה אם אתם יודעים מה זה?
ואז צריך כאילו כולם יוצאים לנחש ומי בגלסים.
אבל מי שיודע יכול להגיד, אני יודע, אני יודע.
הוא יכול להגיד, אני לא יודע, אבל לא להגיד לי.
טוב, אבל להגיד,
They have like 20 different
וגם
columns.
זה
You
העובדה
know where you
שלך,
found
לא,
them?
אני צריכה להגיד,
זה שנה א' של אל. כן, כן. Okay. So, first, if you can start passing this attendance. Can you write the date in the top? Yeah, just the date in the top and your name, and start passing it like that. Okay. So, let's start this over so it will be organized. So, generally, and this is I told you it's not true anymore. Each neuron produces just one type of neurotransmitter, but again, we know nowadays that there are neurons that can co-release two types of transmissions, and the neurotransmitters are always synthesized in the cytosol of the cell, and then are transported into these synaptic vesicles, which are small, 40 to 50 nanometers in diameter, through a two-step mechanism. Okay? So the first stage of the step is that the cell transports hydrogens from the cytosol to the lumen in order to produce a high concentration of hydrogens inside this lumen. And then in the next step, and this is done through a V-class ATPase or V-class pump, and the next step is to use a transporter that harnesses the energy of the concentration difference of the hydrogens in order to move back the hydrogens to the cytosol of the cell and the neurotransmitter into the vesicles. So I know I told you that this is like a form. I always look at it as the cell is first charging these vesicles in energy, and then using that energy in order to load the vesicle with neurotransmitters. But it looks a little bit weird, right? Because we said in the past, I gave you the example of the concentration difference between the inside of the cell and the outside of the cell, and I told you that this is like a form of energy that the cells can use because it's very global, it's very fast. For example, to generate a to generate an action potential that takes 1 millisecond, you can't start transporting ions with ATP. You want something that is much faster. So the difference in the chemical concentration between the outside of the cell and the inside gives the power to generate the action potential. But in this type, like in this example, actually both processes are pretty slow. Like transporters are pretty slow, and also the ATP pumps are pretty slow. So why not just make a ATP pump that just uses the hydrolysis of ATP in order to pump neurotransmitters inside the vesicle? Why do it in two stages? Do you have some kind of thought or intuition to why? I don't know the answer, obviously, but do you have an intuition to why this might be useful? To divide this step to two steps to what?
Maybe you can load many different types of neurotransmitters.
Right. You see, why are you shy? You have good intuition. So first of all, we can get an initial hint from looking at the structure of this thing. Okay, this is extremely complex pump, and actually ATP-powered pumps, they have to like hydrolyze ATP, and they have to have a massive conformational change, and they need to be very specific and stuff like that. And this is something that if the cell or the organism, I told you that there is like 7 or 8 types of neurotransmitters. So if the cell would have to synthesize like a gene, a special gene, or a protein that will be specific to each type of neurotransmitter, then it will require a very high level of complexity. It will require the system to be much more complex than in a state in which you have this like general power loading capability that is pumping like the hydrogen inside the vesicle, and then each type of cell can can express the transporter, which is, I didn't tell you, but I'll show you an example in a second, which is a very, very simple protein. Much, much more simple than this, and also much smaller. And like the operation mechanism here is much more simple than in a ATP-powered pump. And so what is the other, can you think about another intuition? Why you want to couple these two?
Saves energy.
Huh?
Saves energy.
I'm not sure if it saves energy. It might be more efficient in energy, I don't know.
There's a high level of hydrogen from the cell, a way to get rid of that.
It actually doesn't change the concentration of hydrogen inside the cell because these are very, very small. Like the lumens are very small relative to the space, the internal space of the cell. But one intuition is that it might be that these are actually like the necessity or the requirement for like neurotransmitter filled vesicles might be different, or the demand for neurotransmitter filled vesicles might be different in different times in the cell. So the cell can actually store a lot of these vesicles that will be pumped with hydrogens, and only in specific locations in the cell, for example, these can be then shipped to like synapses in the cell, and then only in the specific synapse they can be filled upon demand. They can be filled with neurotransmitter and be ready to be released. Okay. So there are certain, like, we have several. Obviously, we don't know the answer, but we can get an intuition of why the biological system is designed in this way, and also sometimes it doesn't make sense, like how the biological system was designed. But in this case, we can find nice intuitions of that. So we'll give the specific example that we'll get of a neurotransmitter release is through the example of acetylcholine, which is the type of neurotransmitter that we work in our lab. So it's the most important neurotransmitter, a neuromodulator, and the most interesting example, and this is what I'm showing it to you, but this is a general mechanism that is true for a lot of the neurotransmitters as well. So this is just an example of acetylcholine, but it's true for a lot of other neurotransmitters as well. So the basic like production and packaging mechanism of acetylcholine is performed by two enzymes or two genes, or two proteins. One of them is the choline acetyltransferase, which takes acetyl-CoA and choline and produces acetylcholine. And the other one is a transporter that is a proton acetylcholine antiporter, which is in charge of pumping the acetylcholine molecules inside the lumen of the vesicle, of the neurotransmitter vesicle. And in this case, it's interesting because, and this is like a snapshot from a website in which we can see like structures and like genomic sequences of genes. So you see this, the trace here actually describes the gene for chat, so the choline acetyltransferase, and we didn't learn enough about it, but still you can, the thin line here are what we call introns, and the thick lines are what we call exons. So actually if we go back to the structure of a gene, then the thin lines are these introns and the thick lines are the exons. So when this mRNA of this gene will leave the nucleus, it will only have these exons. So it might look very long, but actually you need to imagine that when this entire mRNA is leaving the nucleus, then all these small fragments will be stuck to one another. So this is the only like coding or protein coding region of this gene, and this is very typical. Also, it gives you a nice intuition of the differences in length between introns and exons. And in this example, actually, the second type of gene, the SLC18A3, again, you can understand that this is not from a Drosophila, not discovered in Drosophila because it's a boring name. So this is actually the gene that codes for this transport. Okay? So actually, in this case, inside the choline acetyltransferase choline acetyltransferase protein or gene that produces acetylcholine, you have inside its intron another gene that codes for the transporter. And this gene, for example, doesn't have any introns. It's just the entire gene is encoded like in this sequence, and it only has exons. Like it's a single exon gene. And also we think that this is one of the ways of the system to make sure that if a cell produces chat, it will also produce this protein. Like it will also have, it will also produce, like, out of the intron. It could be that it's independent, but it also could be that out of the intron that is cut out during the production of chat, this protein will also be made. So it can be that an intron from one gene can be an exon for another gene. Okay? So, and this is like a way that this is like a scheme that describes the entire release process. So first we said, and here it looks like it's coupled, but it's actually it doesn't have to be coupled. In which first the ATP powered pump is in charge of pumping hydrogens inside the vesicle. Then a transporter is in charge of pumping the actual neurotransmitter inside the vesicle. Then the vesicles are actually transported to an area of the synapse where they're kept and stored. And we often define two types of population of vesicles in the synapse. We can see that there are some vesicles that are just, and this is like a EM image of a synaptic cleft. So this is like the presynaptic cell, and this is the postsynaptic cell, and this is the synaptic cleft between these cells. And here you can see these vesicles. And you can see that there are vesicles that are docked, what we say like docked to the plasma membrane, and there are vesicles that are free floating and are not docked. And it's believed that these two populations are more or less like independent of one another, and these are like and after the neurotransmitter is pumped into the vesicle, they stay aside, and there is some kind of signal that makes them transfer from to move from being just free floating in the cytosol of the cell and getting docked to the synaptic cleft or to the membrane of the cell itself. Then, after the docking, they wait for a signal, and this signal is actually not an action potential. Okay, so it's not actually that depolarization itself that causes the vesicles to fuse with the synaptic cleft and release the neurotransmitter. What actually causes it is voltage-gated calcium channels. So these calcium channels are affected by the depolarization, like the example that we saw before in sodium channels. So once depolarization arrives to the synaptic cleft, these calcium channels open, calcium enters the cell, and it's the calcium that actually triggers the fusion of these two membranes together, like you can see here. And after this fusion, this this is not really accurate, but you can imagine that it completely fuses, like the vesicle completely fuses with the synaptic cleft and releases the neurotransmitter to the synaptic, or to the space between the two cells. Then there is a process also of recycling these vesicles, and it happens with a very, very interesting protein or a protein complex that's called clathrin. So here you can see like a molecular model of this clathrin. So it's actually like a cage, like a protein cage that stabilizes this structure enough for the membrane to bud off and then it could be recycled again and filled with neurotransmitter for the next round. Okay, so this is a very complex process, and just so like you already know that I don't really like all these cartoons. You know, you get the feeling from here that this vesicle is like everything is smooth and flat, and it's like but actually there are a lot of proteins that mediate all these different activities, like the docking and the release and everything else. And I actually found a nice movie that shows a more real-life model of a vesicle. What? Yes, because they're more complex.
Okay. Vesicle with the membrane lipid. This model represents a cutaway view of a synaptic vesicle with the membrane lipids and proteins drawn to scale. Each synaptic vesicle membrane contains approximately 7,000 phospholipid molecules and 5,700 cholesterol molecules. Each vesicle also contains close to 50 different integral membrane protein molecules, which vary widely in their relative abundance. The most abundant protein is a snare protein, called V-snare. This molecule participates in membrane fusion at the synaptic terminal, and there are about 70 copies of this protein per vesicle. By contrast, the vesicle contains only 1 to 2 copies of V-ATPase. The V-ATPase uses energy from ATP hydrolysis to pump protons into the vesicle lumen. The resulting electrochemical gradient provides the energy to import neurotransmitter molecules, such as glutamate, into the vesicle. Through a transporter in the membrane, protons flow down their concentration gradient to the outside of the vesicle, as glutamate enters by an antiport mechanism. In this way, a vesicle is loaded with thousands of glutamate molecules. This model was created in part from an electron tomogram of a real synaptic vesicle. The tomogram combines visual slices of the vesicle at various angles to create a unified image. Other data, including structural data of the synaptic vesicle's proteins, have been combined with the data from the tomogram to create the three-dimensional model on the left. Note, only 70% of the membrane proteins estimated to be present in the membrane are depicted in the model. A real vesicle would be covered by an even more dense forest of proteins.
So you can see even the most like a very simple structure and very small structure. And also you saw that she started talking about numbers of proteins. Okay, it's so small that we can say that there are 70 proteins. Like, we're not even talking about in the thousands. And even this small structure has so many complex proteins that are on it and responsible for so many functions at level of regulation. So I don't want you to get the feeling that biology is simple. Okay?
Can I ask a question?
Yeah.
Can you go back one slide?
No. Just a minute. Yeah.
Oh, maybe another one.
This.
Yes. So, the pump brings inside the hydrogen.
Yeah.
But it's still, the pH is still lower than outside?
The pH, it pumps, so pH is the minus log of the concentration of hydrogens. So if you have like more hydrogens, then you have a lower pH. Okay? No, this is why this is here. Okay, so after we've released our neurotransmitter, the way that signaling is terminated, or a synaptic signal is terminated, can be either by degradation or reuptake of the neurotransmitter. So in most neurotransmitter, actually, the neurotransmitter itself is recycled. So not only the vesicles, but also the neurotransmitter is recycled. But in the case of acetylcholine, for example, it's actually degraded by an enzyme called acetylcholinesterase, which breaks it inside the synaptic cleft to choline and acetate, and only the choline is transported back to the presynaptic site. So there is also a form, like part of the neurotransmitter is recycled and part of it is broken down, and a lot of the drugs that you know it in psychiatric compounds are actually targeting the reuptake of neurotransmitters. For example, and because, for example, Prozac is an inhibitor of serotonin reuptake. So if it inhibits the reuptake of the neurotransmitter, it means that the neurotransmitter will stay longer in the synaptic cleft, and it will cause its effect to amplify. Okay, so if you have something that blocks the reuptake of something, then you amplify the effect of that neurotransmitter. So again, if we return to the example of acetylcholine, this is the entire biosynthesis of acetylcholine. Actually, acetyl-CoA is a byproduct of the mitochondria, and acetyl-CoA together with the choline that is recycled and is brought in from outside the cell, the chat enzyme that I showed you before produces acetylcholine. Then the transporter is in charge of loading the synaptic vesicle with acetylcholine, which is then released to the synaptic cleft. And different to other neurotransmitter, in this case, after the acetylcholine is recepted by the postsynaptic cell, it is also broken down by an enzyme that's called acetylcholine esterase, and hence it loses its activity. So once it's broken down, it loses its activity, and then the acetate just diffuses out, and the choline is being recycled again for another cycle. So there are two types of, now that we talked about like the synthesis of, synthesis of the neurotransmitter, the transmission of the neurotransmitter and the degradation of the neurotransmitter. Now we'll talk a little bit about the reception of the neurotransmitter, meaning that this happens in the postsynaptic cells. So neurotransmitter receptors fall into two categories, one that we'll talk right now and one that we'll talk in next class because it involves G protein coupled receptors, which we have a separate chapter that's dedicated to this. So you can either have ligand-gated ion channels that open immediately upon binding of the neurotransmitter, and we call them ionotropic channels or receptors. And the second type is G protein coupled receptors, that the binding of the neurotransmitter is not directly coupled to an opening of a channel. It can do various things in the cell, also open channels, but normally it will be much slower if it opens a channel, and the duration can be like a period of seconds to minutes. And in the ligand-gated ion channels, it's normally like millisecond time scales. Okay, so again, if we go back to the example of acetylcholine and we look into inside the example of a muscle cell, so the muscle cell receives, what is it doing? Okay. The muscle cells receive the signal that causes the muscle to contract from a cholinergic neuron, or from a neuron that releases acetylcholine to the synaptic cleft, and this is in turn recepted by special type of receptors that are sitting on the muscle cell itself, which will later cause the muscle to contract. So this is like an EM, a really nice EM image that shows you like the axon terminal, the synaptic cleft, which is like these gaps, and the target muscle cell, and you can see here really in a really nice way like the synaptic vesicles that are filled with neurotransmitter. And this type of structure is actually called the neuromuscular junction. It's a very, very strong synapse and very precise synapse. It's what you have, it's what activates all the muscles in your body. So the type of receptor that's responsible of detecting the acetylcholine in the muscle cells is called a nicotinic acetylcholine receptor, and it's an ionotropic receptor, meaning that the binding of the acetylcholine itself causes a channel in the same protein to open and induce permeability to sodium. It can also be sodium and also potassium, but in this case it's mostly sodium. And this triggers an initial depolarization that will open other voltage-gated sodium channels that will cause a more massive influx of sodium inside the cell, which will later trigger the contraction of the muscle itself. Okay, so release of one acetylcholine vesicle will result in opening of about 3,000 of these ion channels. So it's a very massive activation. And if we go into more detail into the structure of this channel, we can see that this protein is a pentamer that's composed of, in this case, two alpha subunits, beta, gamma, and delta subunits, and the size of it is about 9 nanometers across, and the size of the pore is about 2 nanometers across. So what happens here is that once you don't see here actually the binding site for acetylcholine, but once acetylcholine is bound to here, there's like a gate that opens that allows the permeability, the fast permeability of sodium and potassium ions that can, in this type of a channel, they actually pass together with the water molecules through this channel. So it's a very non-selective channel. The only thing that it's selective for is actually for positive. You see that there's a negative core inside the inside of the protein that actually attracts more positive ions. So the sodium and potassium can freely pass once this channel is open, and the opening is triggered by acetylcholine. So and also there are sites of modulation on this channel that can be, for example, blocked if one of the ways that cobra venom or snake venom acts is by binding to this channel and actually rendering them or destroying their function. Okay, so the binding. This is a model that shows how, like a cobra venom molecule can bind to this channel, and actually this is what causes paralysis in the victim of the cobra. Okay, so with this we end this chapter. Do you have questions about something? Yes.
The last acetylcholine receptor you showed, is this name is another name for the nicotinic, or it's another receptor completely?
This is a nicotinic receptor. It's called the reason that it's called nicotinic is actually like a lot of times the names comes from their discovery. People have found out that when they add nicotine, then these channels become active. Actually, nicotine induces sometimes more activity than acetylcholine, but it's not like. Actually, later there are several types of nicotinic receptors that are not activated by nicotine at all. Like the first channel that they discovered, the receptor for acetylcholine, was probably activated by nicotine, but then all the channels that they found that are similar to it that are activated by acetylcholine were called nicotinic. Okay, but it doesn't mean that they're activated by nicotine actually. It's very confusing, but this is a type of nicotinic receptor. This is actually called like this will be alpha 2, beta 1, gamma delta nicotinic acetylcholine receptor. Okay, so it's a type of ionotropic receptor, like I told you before, that directly couples the binding of acetylcholine. You can actually see here the binding site of acetylcholine. So once acetylcholine is bound, there's a conformational change, and this channel opens to allow sodium and potassium to enter the postsynaptic cell. Okay, so now we'll start the next chapter, which is also a very large chapter, and before we start talking about specifically G protein coupled receptors, again, I feel like I'm obliged to not hide like the bigger picture here from you, which is generally talking about signaling pathways. Okay, so the G protein coupled receptor pathway, which is only one type, is only one type of signaling pathway that the cell has. Cells in general have a lot of, in biology, have many ways to communicate with one another, and there's also many ways that the cell can interpret the signal that is recepted by it from other types of from other cells or from the environment itself. So before I start talking specifically about G protein coupled receptors, it's first we need to do an introduction of intra and intercellular signaling. So intra is obviously between cells, and so this is this part, and inter is what happens inside the cell when the cascade continues, like when the molecular cascade continues. So we can divide like three types of intracellular, of intercellular signaling. One type is what we call endocrine signaling, which is actually signaling molecules that act on the target cells which are distant from the site of the synthesis. So all types of, for example, hormones that are synthesized in one part of the body and recepted by cells in the other part of the body, we'll say that this is a type of endocrine signaling. Paracrine signaling, which is signaling the molecules that affect cells that are nearby. For example, neurotransmission is a type of paracrine signaling. Okay, so it's a subcategory of paracrine signaling, which is again cells that are talking to each other in close proximity. And the last one is autocrine signaling, which is cells that actually respond to substances that they release. So I know it doesn't make that much sense, but again, you can think of biology as something like it's like a cell is like a thinking thing. A lot of times in biology, there are solutions that are found that give like more simple solutions, and this is why we also have like autocrine signaling. Like cells also respond to compounds that they themselves release. So reuptake, I don't know what's the in this type of signaling, we're talking about molecules that make an effect on the cell itself. So I don't think that when the cell is like reuptaking a neurotransmitter, it reacts to that neurotransmitter that it recepted. Other like from what I know, but for autocrine signaling. Yeah, a lot of times you have like it's more complex than that, but a lot of time in like cellular pathways, for example, if a cell is in need of like more energy, it can release some types of signals to the outside of the cell, and then the cell itself, like other compartments of the cell, can detect that and start producing more energy inside the cell. Like it's not. What I'm saying is that it's difficult to look at the cell like it's enclosed and it's like a compartment that's acting on its own. Sometimes the signals, like if biology finds, for example, a solution that if you see a certain type of signal in the cell, then that cell would start producing more energy. Then it's not only signaling that comes from outside. The cell itself can use that same pathway in order to produce more energy. To just release a molecule that will tell the cell itself start making more energy, for example. But I don't want to get too much into detail in that. So just to give you an example of a nice example of how intracellular signaling can cause, for example, this is one of the nicest example of from development, in which like intracellular signaling can help create like biological boundaries. So, for example, one of the mysteries of development is how you know that when we start in early development of life, all the cells are identical, but then there is a process of differentiation. Okay, like cells tend to specify between one another, and also you have compartments. You have organs that are being starting to be made, and you have I don't know, skin and muscle and all these, and they all start from the same type of cells. So, for example, if we imagine that there is some kind of like one cell or part of the cells spontaneously start producing a type of signaling molecule. In this case, it's actually it's known that in the development of the nervous system, there's a molecule that's called sonic hedgehog, or SHH, which you can actually imagine from which type of biologist found it. So again, it's Drosophila people that found it, and I'll show you an image of the fly in a second. But what what they actually discovered is that in the sonic hedgehog molecule, it actually, different concentration of the sonic hedgehog can cause specifications of, like if cells feel different concentration of this molecule, they behave differently. And this is a very nice simplification of how exactly this happens. So if we start from cells that are identical to one another, and then spontaneously one of the cells, like in one of the sides, starts producing this molecule. Actually, it's here. Like one of the cells in this side start producing this molecule, and here you'll find a high concentration of this molecule, and here a low concentration of this molecule. Then the cells will start reacting according to the different concentrations that they feel, and start differentiating to different types of cells according to the concentration that they get. And then the secondary response of these cells will be to start, because these two, there can be competing processes inside the cell that will inhibit one another. For example, let's say that a cell that feels a low concentration of sonic hedgehog will start developing the blue protein, or the blue molecule, and a cell that feels a high concentration of sonic hedgehog will start developing or start expressing the red protein or molecule. And these two proteins have actually a competing interaction between them, meaning that if there is a specific cell that produces more blue than red, then eventually, because there's an inhibition on the production from the blue on the red, only the blue will be expressed in the end of the cell. For example, if we take a cell here that has more blue than red, then in the end, the entire cell will be blue and will not express the red protein at all, and the other way around. So this is like the way that the simple gradient in concentration can cause like initial specification. Then there can be like fine-tuning, and then secondary interactions and secondary concentration that can later cause the differentiation of this tissue to different compartments. So just to show you like the sonic hedgehog, the sonic hedgehog fly, and the reason that it got its name is that this is like a normal Drosophila. Okay, so you see it has like a nice structure, normal sized hairs, not like that you're expert in Drosophila, but this is how the sonic hedgehog Drosophila looks like. Like if this molecule or this protein is mutated or cut out of this, so you can see that it has severe developmental issues, and also it looks like a hedgehog. So from that the name, but actually this molecule is very important for development in all, also in mammalians and other organisms. So this is the level of the. So this is just an example of like what can intercellular signaling or communication between cells in development cause like biological boundaries and specification to different types of cells. And now we move on from the level of the molecule itself, which I give you like the example of the sonic hedgehog, to the level of the receptor. And in the level of the receptor, so each receptor binds only a single signaling molecule or a group that is very closely related. But in contrast, the signaling molecule binds to multiple types of receptors. For example, acetylcholine can bind to different types of receptors, like and induce different types of signaling pathways and reactions. But the receptor itself is normally very, very specific to the type of molecule that it recepts. Okay, so you can look at it as for the cell, the identity of the signaling is extremely important. But the body can use the same molecule to send different signals to different cells. Okay, so acetylcholine, for example, can cause, if it's recepted by a muscle cell, it will cause contraction. If it's recepted by heart muscle cells, like this is periphery muscle cell, it will cause contraction. If it's recepted by heart cells, it can cause like slower heart rate. and if it's recepted by the pancreas, it can cause the release of digestive enzymes, for example. Okay, so there's different specifications, different activities of different molecules. And also, this example is here to show you that although in this example it looks like the receptor is much larger than the molecule that it's recepting, it's not actually the case at all times. For example, this is a molecular model of a growth hormone that is a signaling molecule, and this is the receptor that recepts the growth hormone. So you can see that the sizes are not always that the receptor is much larger than the signaling molecule itself. A second type of phenomena that is very common to signal transduction is that although there is high degree of specification, I told you that the receptors themselves are very specialized. They're very specific to the type of molecule that they recept. But actually, the end result, when we look at it inside the cell, is often results in the same cellular response. For example, it's very common that in G protein coupled receptors, like we're going to talk in the rest of this chapter, it's very common that it doesn't matter which type of molecule will activate the G protein coupled receptor. In the end, the response of the cell can be will a lot of time be an elevation of a molecule that's called cyclic AMP. Okay, so again, this is one of the things that it seems like it doesn't make so much sense, because the cell is put so much energy into to knowing exactly what is the molecule that it recepted, but in the end, the bottom line is that the molecule that is affected is a lot of time the same molecule. Okay, so different types of signaling molecules can induce a cascade that is very similar. And don't worry if it sounds very confusing. It's just we'll go over it next class in more detail. And also, this is like a general introduction to signal transduction, so I'm speaking like in the air. So focusing on so up until now we talked about the molecules that are being recepted by the cell, the receptors, and now we'll say a few words about the activator molecules, or the molecules that actually do the work inside the cell after the signal was recepted. And these molecules are actually a lot of time called first messengers, because they are the first molecules that are produced or are responding to the signal that came from outside the cell. So the ligands are actually the first receptors. The first molecules are the first messengers, and the second molecules that react inside the cells are called the second messengers. And the second messengers can cause a lot of different types of effects inside the cell. For example, second messengers can be one of the most common messengers is a cyclic AMP, which is a derivative of ATP, but in this case, instead of having it's adenosine with a base, but it has, instead of having 3 phosphates, it has this cyclic conformation. So the phosphate here through the oxygen is bound to the sugar backbone. So this is a very common type of secondary messenger. But there can also be calcium is also a very typical type of secondary messenger, and we also see that it can have different functions in different cells. For example, in muscles it causes contraction, and in nerve cells we saw that when you have a signal it can also cause exocytosis and release of neurotransmitter to the synaptic cleft. So it has different functions in different types of cells. So I'm thinking about maybe how much more do I have here? No, I think we'll continue with this next class. Also, we'll do it in a more ordered way. But still, next class I want you to read a paper. There's a very classical paper by by Kandel, by Eric Kandel, which is one of the